Welcome to part 2 of the lecture on chromosomal variations. What we're transitioning to in this part of the lecture is dealing with variations that occur typically to individual chromosomes. So the first example that I want to go over is a simple deletion. Now, a deletion again is a pretty straightforward concept, and that is a portion of the chromosome has been deleted or missing somehow. So if you look at the left-hand side of this figure, you can see a chromosome with genes A through G listed on the chromosome structure. And in the second half of that figure, you can see that genes E and F are missing from the chromosome and, in a sense, have been deleted overall. How do these types of deletions take place? Well, the easiest example is what's known as a terminal deletion, which just means that the end of a chromosome has been broken off and has been lost. Now, there are lots of repair mechanisms in place to repair chromosomal damage, and oftentimes when breakage takes place, it does in fact get repaired, so the little piece could be essentially glued back onto the main chromosome. But if that does not happen, that portion that has been broken off is lost simply because it does no longer have a centromere association. If you think back to both mitosis and meiosis, the centromere is where microtubules attach and specifically allow for the movement of the chromosomes during either one of those nuclear division events. The piece of the chromosome that has been broken off no longer has a centromere for microtubules to attach to and hence doesn't get moved during the process we've just discussed. Another way that this can actually take place is referred to as an intercalary deletion, and this is an internal deletion that takes place. Chromosomes themselves are not hard, fixed, rigid structures. They are soft, pliable, bendable structures. Think about them sort of like a piece of spaghetti noodle that's been cooked. So they can move around in all kinds of different shapes and structures. And what can happen in one circumstance, like you see in this slide, is that the chromosome has looped around on itself. This does cause a little bit of stress and can cause breakage of the chromosome. Now, in many circumstances, like I mentioned, there are repair mechanisms that can fix this problem. However, if the repair mechanism doesn't repair properly, and instead of putting the piece back in that needs to be put in, it just omits it entirely, sort of like a cutting and pasting type of situation. What happens in these situations, again, is that a portion of the chromosome is lost because it no longer has the centromere association required for the microtubule attachment for chromosomes to move during mitosis or meiosis. The next example of chromosomal abnormality is what's referred to as a duplication. Here too, a straightforward concept in that you have a portion of the chromosome that is repeated or duplicated. In the figure you see genes E and F in particular in the top panel, and in the bottom panel of the figure you see that genes E and F have been copied or duplicated overall. How exactly does this take place? Well, it is actually problems associated with genetic recombination or crossing over. If you think about what genetic recombination or crossing over is defined as, it is a reciprocal exchange of genetic information on homologous chromosomes. And the key there is a reciprocal exchange, meaning you have a piece that's broken off of one and a piece that's broken off of the other, but it's the same amount of genetic information that gets swapped during the crossover event. If that doesn't happen correctly in what's referred to as unequal crossing over, you have two situations arise at the end of that event. you have one chromosome that has lost some information and one chromosome that has gained additional information. These gene duplication events are actually very important evolutionarily. I'm going to give you two examples in the next slide, this slide and the next slide. So the first example is an example of duplication and divergence of genes in the hemoglobin family. Up at the very top of this figure, you see what's referred to as an ancestral globin gene, where there was then, in the next step, a duplication event that took place. After the duplication, there was sequence divergence in these two genes, producing what's known as the alpha and the beta form. Then from each one of those, there were additional duplication and divergence events, obviously multiple as you work your way down this particular figure. But what this results in is actually what's known as a gene family, where there are multiple genes that are obviously related in some way because of sequence, composition, function of the product, and that type of thing. And these are all typically due to duplication and divergence events. This is another example of evolution in action with duplication and divergence of genes. In the left-hand panel, you see a wild-type chromosome with genes A, B, and C. In this example, the expression of those genes and the protein products in particular interact in a specific way during embryonic development in this fish. This results in the developmental pattern and the striping pattern that you see on the fish in the panel on the left. In the panel on the right, you see a duplication of gene B has taken place. So instead of a single copy of gene B, there are two. In this case, there can be divergence in those two genes. However, what we are looking at in this situation is, again, the interaction of those gene products once they are produced. And during embryonic development, those gene products interact in a different way now because there are different protein products present and produce a different phenotypic effect on the fish. We don't see that striping pattern like we see on the left-hand side, but more of a spotted pattern overall. And this, again, is a documented case of duplication and divergence. The next example of chromosomal abnormality is what's known as an inversion. Just a few slides ago, I mentioned the fact that chromosomes are not hard, fixed, rigid structures. They are soft, pliable, flexible structures. And as such, these chromosomes can form loops like you see in the bottom panel of this particular slide. Now, like I mentioned before, when we talked about deletions and these such loops, in that this does cause stress and strain on the chromosome, and oftentimes damage can occur where there's a breakage at the looping point. Because there are repair mechanisms that come in and oftentimes fix these problems, they go undetected. However, in this example, if the repair has occurred improperly, so there has been a repair, but the repair is not pasting the pieces back in the same way, however, flip-flopping them, and that's really clearly seen again in this bottom panel of this figure, you can see that the result is what's known as an inversion. So in the top panel, you have genes A through G, and the inversion has resulted in D, E, and F being in the opposite orientation overall. Now, inversions can be categorized as paracentric or pericentric, and this really is just a definition that defines or describes where the inversion has taken place. If it includes the centromere, that's referred to as pericentric. If it does not include the centromere, that is referred to as paracentric. The way I always remember it is para with an A avoids with an A the centromere, And peri, with an I, includes, with an I, the centromere. So again, just a specific designation for that. The next chromosomal abnormality I want to discuss is referred to as a translocation. A translocation is where you have one portion of a chromosome moved to an entirely new location. So this is not the same as genetic recombination, because genetic recombination occurs on homologous pairs of chromosomes. This is a situation where you have genetic information from one chromosome moved to a completely different chromosomal location overall. This is in fact what happens when we discussed Down syndrome in the previous, or part 1 of this lecture, where we had a small percentage of Down syndrome cases due to translocation. So in this figure, what you're looking at is a chromosome that has genes A through G, and a separate distinct chromosome that has genes M through S. These are not homologous chromosomes because they have different genes located on them. They are in fact 2 distinct chromosomes overall. In this example, the translocation is where you have genes E and F and genes Q and R basically flip-flopped from one another. Okay, so in this figure, in the top portion, panel A, you can see how this type of translocation could possibly take place. Again, chromosomes not hard, fixed, rigid structures, soft, pliable, flexible structures. Again, think spaghetti noodles, cooked spaghetti noodles. And so what you see in panel A, the first portion, is these 2 chromosomes, again, not homologous, happen to just come in contact with one another or come very close to one another. If there is in fact breakage that occurs of each one of these chromosomes, and those repair mechanisms that I mentioned before do not repair properly, you could see where a situation might arise where there is a swapping of genetic information, in this case, between these non-homologous chromosomes. Again, this is not the same as genetic recombination. This is a distinct mechanism where you have breakage and repair, but the repair occurs improperly. Okay, so why is that a problem? Well, when you have homologous chromosomes pair up during meiosis, this is where the problem comes into play. In panel B, you can see that the homologous portions of these chromosomes are pairing up. So for the blue chromosome, for example, you can see gene A, gene B, gene C pair up very nicely. You can also see the orange portion, gene O, gene N, gene M pair up very nicely. But because there has been this translocation, you get this very odd structure that occurs during synapsis. And again, synapsis is when homologous pairs of chromosomes align or pair up. So what does that mean?